Why genetic codes are used in replication of protein. If amino acids and mRNA are directly related, amino acids connect selfishly, so proteins cannot be replicated accurately. A tRNA carries an amino acid to mRNA together with a genetic code. If the genic codes between tRNA and mRNA are corresponded, the amino acid is set to the position specified by the mRNA. There is two strands in a double helix of DNA. One strand is a series of molecules with right-handed symmetry, and the other is a molecular sequence with left-handed symmetry. The molecules those are elements of a helix do not connect with the molecule with different chirality. Each molecular sequence possesses different function. Why molecule of control and the molecule to be controlled has different chirality. Almost all living things on Earth, composed of sugars with right-hand system of D-type, and the amino acids with the left-hand system of L-type. The molecule to control and the molecule to be controlled are adjacent and activated concurrently. L-typed amino acid and D-typed tRNA are adjacent and simultaneously activated and interact with each other. So, tRNA carries an amino acid. How an amino acid and glycans join in tRNA. The D-typed swirling water region occurs at the neighbor of a L-typed swirling amino acid swirl. A string of long-chain carbohydrates containing D-type monosaccharides produced on a surface meshes by opposite rotational directions. The produced transfer RNA carries the connected amino acid. The messenger RNA that conveys the sequence of amino acids is generated in the vicinity of tRNA. Since chirality of mRNA is L-type, mRNA and tRNA contact at one point as a pair of gears. mRNA is not related to amino acids unless it is mediated by tRNA. The position of tRNA in the sequence of mRNA is determined by matched genetic code. The anticodon of gene code of tRNA is checked to the codon of the genetic code of mRNA in a unit of amino acid. When codons and anticodons satisfies the matching conditions, amino acids carried by tRNA are incorporated into proteins. The tRNA with an anticodon that had detached amino acid is able to carry the same amino acid again. Key lock complementary relationships on codons of mRNA and anticodons of tRNA. The genetic code is composed of 3 sets of 4 bases. 4x 4 times 4 equals 64 types and is associated with the 20 amino acids that make up a living body. Key lock complementary relationships is different from enantiomer as those have the same molecular formula and structure, but are asymmetric carbon atoms, and they mirror each other and do not overlap. Hydrocarbon molecules that form cell membranes of living organisms. Molecules of the cell membranes of living organisms suspended on surface of water. The cell membrane of current organisms consists of the hydrocarbon molecules C16H34 or C18H38. In the primitive Earth, CO2, which was the main component of the atmosphere, collided with H+, and many of the hydrocarbon molecules synthesized in the sky remained in the sky because of their low boiling point, and the chemical reaction had proceeded. However, C16H34 had a melting point of 20 degrees Celsius and a boiling point of 300 degrees Celsius, and C18H38 had a melting point of 27.8 degrees Celsius and a boiling point of 316.15 degrees Celsius, so it became a liquid and floated on the surface of the water. Molecules and tissues of intermolecular bonds are repeatedly produced and extinguished. The change in free energy when hydrogen and oxygen atoms form covalent bonds is -50 to -110 kilocalories per mole. The energy of hydrogen and ionic bonds is in the range of 3 to 7 kilocalories per mole. The binding energy of the van der Waals bond is 1 to 2 kilocalories per mole. The energy of thermal motion at 25 degrees Celsius is 0.6 kilocalories per mole, but the range of energy of thermal motion is large, and there are many molecules with kinetic energy that break the energy of hydrogen bonds and ionic bonds. Formation of cell membranes and thermal vibration in helix structures of a liquid water. Formation of cell membrane. Liquid water occasionally forms a helix structure. It possesses penetrating vacant shafts and molecules of the oil film penetrate into the vacant shaft perpendicular to the water surface. Fatty acid molecules with a carboxyl group COOH attached to the tail form cell membranes perpendicular to the water surface due to hydrophobic interactions. The hydrophilic part will be set at outside in the cell membrane. These fatty acids are found in large quantities in natural fats. Motion of molecules on the surface of water. Carbohydrates CXH2OY are produced from hydrocarbon on the surface of hydrocarbons floating on the surface of water, and molecules of polysaccharides containing number of monosaccharides are produced. There are upward and downward thermal motions of adjacent molecules in the helix structure of water molecules. Generation of tRNA linked to amino acids. A long chain of carbohydrates consisting of D-type monosaccharide units that swirl at a point in contact with the invaded L-type amino acid region is added. The molecular group of D-type sugars that bind the amino acid units together became the D-type transport RNA when making proteins. Generation of biomolecules by cell membranes. When L-typed amino acids invade the cell membrane, the same L-typed amino acids invade in succession. When amino acid molecules of the same chirality bind peptide, helix-like and ribbon-like proteins are generated, which easily detach from the cell membrane. A long chain of carbohydrates containing D-typed monosaccharides and forms a thread of molecules that contains D-typed monosaccharides at the surface. Amino acids with L-type chirality and the D-typed nucleic acids are produced almost concurrently. And D-typed swirl region neighboring L-typed swirl region rotates in opposite direction via Geer's mechanism. Mechanism of tRNA generation by L-type amino acids and D-type glycans. The scenario that DNA that stores nucleic acid sequences was first generated at the origin of protein-making system cannot be accepted. At first, tRNA was produced, and production of mRNA followed. DNA was generated using mRNA as a template in the early stage. Later, RNA is made from DNA according to central dogma after evolution of mechanism that is needed helicase and polymerase. Configuration of double helix structure of DNA. When a protein is released, D-type tRNA with anticodons and mRNA with codon remained. Molecular substitution takes place in the process of generating DNA from mRNA. The sugar contained in RNA is ribose, and the sugar contained in DNA is deoxidized deoxyribose. Thymine, the base contained in RNA, is uracil in which CH3 is replaced with hydrogen in DNA. In the structure of DNA, the direction of the shift of the nucleotides of the two stringy molecular rows is opposite, but there is no problem because they are converted into RNA and replication takes place at one point of work. Production of DNA by using mRNA as the template. Divide the double strand of DNA with hydrogen bonds of the bases. A single leading strand corresponds to mRNA. As for the lagging strand, the remaining chain molecular sequence is intermittently inversed and it connect the tRNA together with anticodons that have finished carrying amino acids. By adding helicase and polymerase, DNA can be replicated from DNA or RNA can be generated. First, the hydrogen bonds of the DNA bases are divided by helicase to form two single strands. Leading strand corresponds to mRNA. DNA polymerase mediates replication of DNA through adding free nucleotide. The remaining single-stranded lagging strand corresponds to tRNA and produces free nucleotide. How to replicate protein by using DNA. DNA is used to generate RNA, and the genetic code is used to sequence amino acids one by one to replicate proteins. Summary. One of the most difficult and unsolved questions about the origin of life was the relationship between the genetic code of DNA and amino acids. In almost all living things on Earth, chirality is made up of proteins composed of DNA, which is composed of D-type sugars, and L-type amino acids. Molecules with different chirality have selective reactions among concurrently activated molecules in replication of protein. That is, D-type system of sugars control L-type amino acids. If amino acid molecules are directly related to messenger RNA, amino acids bind selfishly to messenger RNA and accurate replication is not possible. Therefore, the relationship between tRNA and mRNA is checked by the genetic code that is not related to amino acids, and the amino acid sequence is determined through tRNA only when the matching conditions are satisfied. Thank you very much for your kind attention.